Okay, so the electron carriers, I'm going to draw here. So you had, out of this, we had, remember, NADH coming to the electron transport chain. And yesterday we drew and talked about how the electron transport chain is a series of proteins and protein complexes in the cristae or the inner membrane of the mitochondria. So you have some NADHs coming to the electron transport chain from glycolysis. We have NADHs coming to the electron transport chain from the intermediate steps. So there's NADHs coming from there. And lastly, we have NADHs coming to the electron transport chain from the citric acid cycle, but we also have FADH too. And so the purpose of all these first steps is to oxidize glucose, and then the electrons that were once part of glucose are in these electron carriers, and they bring them to the electron transport chain. So then once they go to the electron transport chain, so we talked yesterday about how the electron, the first bullet, the electron transfer in the electron transport chain causes proteins to pump H pluses from the mitochondrial matrix. So let's look at this. Let me change colors here. So when the electrons from the NADH, what happens is the electrons go through the electron transport chain, kind of like this picture here yesterday that we talked about. So the electrons go through from molecule to molecule. And when I put these big green guys up here, these are those big, large protein complexes right here, made out of smaller molecules. So then the electrons move from molecule to molecule in the electron transport chain. Why? Because each molecule is more electronegative than the next. So then we end up with these electrons, these two electrons meeting up with H plus and O2. So these two electrons that are they're moving through, they came from NADH, the two electrons, they move through. And these two electrons now meet up with two H pluses and a half of an O2, and what we get is H2O made. All right, and so that's your waste product there. So why in the world then do we need these electrons to move through these electron transport chain? What is it doing? Remember this, we're trying to make ATP. So the electrons moving through the electron transport chain causes the proteins to pump H pluses from the mitochondria. So remember that the pump means that it uses energy. So we talked yesterday about how there's a high H plus concentration in this intermembrane space and a low H plus concentration in the matrix. And so what happens is when these electrons go through this protein complex, it pumps an H plus across. Then when the electrons go through this complex, it pumps an H plus across. That energy from the pumping doesn't come from ATP, it's because of the redox reaction. So remember that when the molecules in the electron transport chain get the electrons, they become reduced. And when they give them to the next guy becomes reduced and the other guy becomes oxidized. And so we have a series of redox reactions through here, and redox reactions are exergonic and they release energy, and that's the energy to pump the H pluses into the intermembrane space. So if we continue following the flow of electrons, when they go through this complex, it also pumps an H plus. And so we have H pluses becoming higher and higher concentration in this intermembrane space. Why is it that we need a high concentration in the intermembrane space? Because that's what the job of the electron transport chain is. That's all it does, is pump H pluses into this intermembrane space. How does that generate ATP? This has to do with the second molecule we talked about yesterday, ATP synthase. ATP synthase is a molecule, it's an enzyme in the intermembrane of the mitochondria that's not part of the electron transport chain. So I'm going to put it over here. Kind of has this shape here. This is ATP synthase. And so this is our enzyme that's going to make ATP. How do you make ATP? You need an ADP and a phosphate, right? We put them together and make ATP. So this enzyme, guess what? As it's sitting right now in the intermembrane space, it doesn't have the right shape. Is that a big deal for an enzyme?
Yes.
Yeah, because enzymes, the active site has to be in the right shape. So in this case, the substrates, ADP and a phosphate, can fit into the active site, and then the enzyme will speed up the reaction of putting the phosphate back onto ADP to make ATP. So right now, this isn't the right shape. So what makes this in the right shape? The flow of hydrogen ions through it. So notice here we said yesterday that we have a flow of H pluses. They go through, the H pluses move back across the membrane. So we just pumped them here, so we have this really high concentration of H pluses. Now, the H pluses are going to go back through and back into the matrix. What happens is that H pluses going through that enzyme changes the shape of the enzyme. And now, the substrates, the ADP and your phosphate, your inorganic phosphate, can go to the active site of the enzyme, and then the enzyme converts that into ATP. And this can only happen when the enzyme is in the right shape. The right shape can only happen when H pluses diffuse through it, and the H pluses can only diffuse through it if there's a high concentration of H pluses here. So therefore, the electron transport chain's job is to constantly pump H pluses here so that those H pluses can move back in, so the enzyme can be in the right shape, so you can make ATP. Does that make sense? So can you make ATP without the electron transport chain?
No.
No. All right? And so these guys couple together. They both have to happen in order for ATP to be made.
Okay, so the H pluses attach to the ATP synthase, and as they're like pumping it back into the mitochondria.
It's not pumping, it's this, look at here, high to low, so that's diffusion.
Okay,
No
diffusing,
energy here.
Okay, diffusing into the actual mitochondrion itself. It changes the shape of the ATP synthase,
Yes.
which
Just the movement of the H pluses through it
okay,
changes.
and then the change of shape creates the ATP.
That creates the enzyme to have the right active site so that ADP and a phosphate can actually get into the active site so that it can create ATP. Yeah, absolutely. And that's what this next picture in your notes shows you is this, what you're looking at here is kind of like what I just drew here. You're looking at my orange here, the ATP synthase, that's this guy here, embedded in the inner membrane. So here's the inner membrane space, so that's this part here. High H plus concentration in the inner membrane space, so you're going from high H plus concentration to low H plus concentration. And so this is diffusion, so there's no energy here. And so when these diffuse through, it changes the shape and it gives you specifics as far as how, as a stator anchored in the membrane that holds the knob stationary, and then as the H pluses, it spins and so on. You don't need to know the logistics of it. What you need to know is just that it changes shape. All right, you can know the logistics of it if you'd like to. All right, but that's not, you won't be tested on that. It's not on the AP test. So the H pluses diffuse through, changes the shape of the enzyme, so that now ADP in your phosphate, remember inorganic phosphate means it's not attached to anything. These guys are your substrates. And the substrates now can fit into the active site, and the enzyme can convert your substrates to your product, which is your ATP. And that's how that works. And so these two together have to occur together, and they collectively are called oxidative phosphorylation. This is called chemiosmosis. This is the electron transport chain, and collectively they're oxidative phosphorylation. Alrighty, so if we look at the next slide here, so the energy stored in a hydrogen ion gradient across the membrane couples the redox reactions of the electron transport chain to ATP synthase. And that H plus gradient is referred to as a proton motive force, emphasizing its capacity to do work. So we have this, what the electron transport chain creates this gradient. Remember we talked yesterday, what is a gradient? A difference in concentration across the space. So in this case, it's across the inner membrane. So you have high and low. The electron transport chain creates that gradient. So therefore, it pumps the H pluses here. And then this is like energy, it's like water behind a dam. You have a high concentration of H pluses in this intermembrane space. So now they can flow back in through the ATP synthase, changing the shape of it and allowing it to make ATP. And so if you didn't have the electron transport chain, what would happen is that you wouldn't have H pluses pumped anymore, and the high concentration of H pluses, they would diffuse through ATP synthase, but eventually would you think the H pluses would reach equilibrium? Yeah, they would if you didn't have the electron transport chain. And if the H pluses reached equilibrium in both spaces, you're never going to have H pluses moving through ATP synthase. ATP synthase won't be in the right shape, and therefore you won't be able to make ATP. So the electron transport chain is very important in order so that you always have a situation where H pluses are going to diffuse through. So this picture here shows you kind of what I just drew up here. All right, and so what you're looking at is, if you look here, this is the inner membrane. So that's why I put him brown. The green guys here in my picture are the major protein molecules, complexes in the electron transport chain, right here, and right here. So these guys right here comprise the electron transport chain. Here's your ATP synthase, not part of the electron transport chain. These two things together comprise oxidative phosphorylation. And so to kind of give you the big picture for like this here, it says that this is the inner membrane. Here's your matrix inside, and here's your inner membrane space. I'm going to go ahead and draw just to show you that there's the outer membrane, so that you can see that these H pluses are between the inner and outer membrane, like we're seeing here. So it's like I'm taking this part of the picture and enlarging it, all right, and what we're looking at. And so I'm just going to color code and kind of go through this on this particular picture. So if you're a color coding kind of person, you might want to get out some things. NADH. So NADH carries electrons from food that was in your glucose molecule. And so NADH carries 2 electrons. So what it does is drop those 2 electrons off at the beginning of the electron transport chain. This arrow here, what this arrow is showing you is the path of electrons. So this is the path that the electrons take. So notice here also, when the electrons move through that first complex, this is a series of redox reactions. There's smaller molecules in here, so the electrons are being passed from molecule to molecule. Energy is released. That energy is used to pump the H pluses into the inner membrane space. So I'm going to write here that these guys are pumped. That's requiring energy. The energy is from the redox reactions. So then the electrons go to this little guy here. The 2 electrons are now here. This is like kind of a shuttle between the 2 complexes. So then it comes over and drops the electrons off, and it moves through the second complex. When that series of redox reactions occurs, it releases enough energy to pump yet another H plus into the inner membrane space. Then those 2 electrons are another little shuttle here, and eventually that drops it off and it moves through the next complex, which then, as it goes through those series of redox reactions, enough energy is released to pump H plus, another H plus here. And then those 2 electrons are at the end of the electron transport chain. Here's your oxygen, which is your final electron acceptor. And remember, the 2 electrons meet up with H plus and form 2 H's and a half of an O2, which is an O, and we get those electrons now are part of water. And so, so that's our electron transport chain. The purpose then was to make it so that we have our high concentration of H pluses in the inner membrane space. So electron transport chain, electron transport and pumping of protons, which are H pluses, which create an H plus gradient across the membrane. So that's what we have established. So then here's our ATP synthase. And so our ATP synthase, the H pluses, now if we notice here these little dots here, all the H pluses, this one, this one, this one, all right, come over here, and they move back into the matrix. Does this require energy? No, because we're going from high to low. This is, I'm going to put here, diffusion. High to low requires no energy there. So what does the H plus do to the ATP synthase? What does the H plus do to the ATP synthase? Changes its shape, right? So I'm going to write that here. So ATP synthase changes shape when what? What causes it to change shape?
Hydrogen ions are present.
When the H plus diffuses through. Alright, so what does it cause to happen? Allowing what to occur?
ADP
Allowing ADP in a phosphate
to go into the active site,
to go into the active site.
and the enzyme can do its thing.
And what is its thing? Go into the active site and be converted to ATP. Very good. So what Max just did in verbalizing it is what you need to be able to do, all right? So because something like this, a lot of people might look at this picture when I'm going over it. Well, yep, that makes perfect sense. And then as you sit down tomorrow afternoon and try and verbalize it yourself, you might figure out that, wow, I can't explain this at all. It really made sense. I thought I understood it. So that's a tactic that you should be doing, like looking at a picture and things like that. Can I verbalize it myself? If you can't, then you don't really know it. All right, so this is the ATP synthase changes shape. Here's our substrates, ADP and the phosphate. And now we get our ATP. And so the two processes, this is chemiosmosis and electron transport chain, occur together. Now I want to point out that there's another molecule that's an electron carrier, FADH2. So FADH2 drops its electrons off here. So here's FADH2. So it also carries 2 electrons. And so the 2 electrons get dropped off here and then follow the same path as the rest of the electrons that were once part of NADH. The only real difference is the electrons of FADH2 never go through this first complex here. All right, and so therefore, there's less H pluses pumped through. Does that make sense? All right, so here there's only 1, 2 pumped out per per FADH2. For NADH, there's 1, 2, 3. So what does that matter? It matters in the amount of ATP that's made. So with NADH, we have 1, 2, 3 H pluses pumped through as they go through these complexes. That means 1, 2, 3 H pluses can diffuse through ATP synthase to make ATP. So therefore, each NADH, since it pumps 3 H pluses, can make how many ATP do you think? 3 ATP. So I'm going to write that here. So each NADH molecule can make 3 ATP. Using the same logic, how many ATP molecules can FADH2 make?
2.
2. All right, because there's only 2 H pluses pumped. So each FADH2 can make 2 ATP. All right, so the next question I'm going to ask, but you have a question, Roman?
You say that each NADH can make 3 ATP and definitely can make 2. But what if there are, I'm pretty sure that there can be only 3 proteins and like every electron. Can it be like, each molecule generate more than 3 ATP?
Each NADH?
She's saying, is the electron chain ever longer than this? Ever has
No,
more
this is
proteins?
it for the electron.
It's always
chain.
3
It's-
proteins.
It's like this, yes. All right, so therefore, one NADH molecule can only drop it off at the beginning of one electron transport chain, which this doesn't change, all right? But what can make us make even more ATP, remember we talked about the purpose of the folds? Because what happens is that the purpose of the folds is to increase in, surface area, so you can have lots of electron transport chains in here. So you know what I mean? There's not one electron transport chain, all right, for a mitochondria. Does it make sense? So you can have lots of them. And so my next question to you is, with this, is that this is the whole process is oxidative phosphorylation. What I'd like you to do is to calculate for one glucose molecule entering the cellular respiration process. I want you to calculate what we put here and how many ATPs are made from this step. We did the first two steps here. How many ATPs are made from this step? What information do we need to know to be able to answer that?
How many NADHs?
Yeah, how many NADHs and FADH2s are coming to this step? Where are they coming from? The previous step. So glycolysis, that intermediate step, and the citric acid cycle. So what I'd like you to do is go back and look at glycolysis, the intermediate step, and the citric acid cycle, and figure out how many NADHs and how many FADH2s are actually coming to drop electrons off here.
Oh, no, ATPs, because I see 1, 2, 3, like directly. 4, 1, 2, 3, 4. And then 1 FADH. Oh, but more is produced. Okay. So in the citric acid cycle, how many are produced? Okay. And there's even more than that. Well, it would be 6, 12, it would be 6 times 3, and then FADH times only 2, because they can only make 2 ATP, don't combine.
Alright, so I'm gonna figure out.
I thought you had it all.
Almost
I
there.
so here you copied my notes from yesterday, so I don't have any of it written down.
Somebody wanted to put you, have to carry him around. Oh, was that really a plan? Yeah. Oh, really?
I probably counted them twice. I got
Alright, how many H plus or NADHs come out of glycolysis?
I know.
36 and 32.
Oh, okay, so you're talking about this here.
Oh, out of glycolysis?
NADHs,
2.
2. And Shane, the fact that you answered 36 to 38 tells me that you looked somewhere. You didn't figure it out yourself. You just gave yourself away there. Alright, so then NADH from the intermediate step?
One. Just one.
It looks in the intermediate step, remember that intermediate step, there's 2 pyruvate, so that happens twice.
So 2.
So therefore we have 2 of those coming from the intermediate step. And then the citric acid cycle happens twice. There's 2 turns of the cycle because 1 for each acetyl-CoA. So when we totaled these yesterday, the picture actually shows 3 NADHs coming off, but since it happens twice, we have 6 NADHs and 2 FADH2s. So now, what does this mean? So, how many NADHs total do we have? We have 2, 4, and then 6 gives us 10. So we have a total of 10 NADHs coming from the oxidation of glucose there. They go to the electron transport chain. Each makes 3 ATP, so we get a total of what?
30.
30 ATP. Then, each FADH2, we add 2 of those. The only place it comes from is the citric acid cycle. So each of those can make 2. So 2 times 2, we get 4. So our total here is 34 ATP. So we're going to write here 34 here. All right, so this is what I mean by 2 ATP, not a lot. And so the whole process of cellular respiration, we're taking into account all the steps. We have 34 from oxidative phosphorylation, 2 and 2 gives us a total of
38.
34, 35, 36, 37, 38, for a total amount for the whole process of cellular respiration. So you can see 34 out of the 38 come from this last step. That's why this last step is so important. But the first steps are important to get the electrons over here so the last step can happen. So you have a bullet underneath here that says during cellular respiration, most energy flows in this sequence. And it gives some blanks there. So we're talking about the flow of energy from the start of cellular respiration to the end. The start of cellular respiration, where's your energy at and what molecule? Yeah, glucose. So for everybody, that first blank should be glucose, right? That's where your energy is. Where's the energy that was once in glucose at the end of cellular respiration? In what molecule?
ATP.
ATP, right? That's where our energy is. So your last blank is ATP. What I'd like you to do is see if you can fill in what you would put for the other blanks. Now this is in general, so we're not there's not enough blanks there to put every intermediate. So you can't say, oh, glucose, then energy is in pyruvate, and then it's in, and so there's not enough blanks here. So in general, where do you think the energy is tied up that was once in glucose as you go along? So see if you can fill those blanks then.
Okay.
That has not been working. I'm sorry, it doesn't work. So you either have to use that one, or I have, some people don't like that one, so I have this one that's kind of broken. It's the worst. You like? I haven't tried that one, but a lot of students
The only thing I can think of is if the first step is glycolysis, it's glucose. And then we have this next step, could it be like citrate? And then the next step could be like phosphate, but that wouldn't make any, like...
No, it could. Well, I see what you're saying.
I want to say that if I would acetyl-CoA and citrate and then to NADH and SODH quick up after that and carry the energy.
Alright, let's see what we can put in this blank. So the energy at the start is in glucose. And then for the first couple steps, remember the whole point is to oxidize glucose to strip the electrons, and remember the carbon and the oxygen are released as CO2. So all the energy that was once in glucose is now where the electrons were stripped from it and are now in molecules of what? The electron carriers. NADH, and we also in that same blank, there's another electron carrier, right? It's FADH2, so we could put NADH and FADH2 in there because FADH2 has some electrons, and therefore these electrons represent the energy. Because then what happens next, those electron carriers drop the electrons off of the electron transport chain. This is energy because what's going on is as those electrons move through the electron transport chain, remember you have a series of redox reactions that's going to release energy. So now the energy is now in those electron transport chains in that series of redox reactions. So like the electron transport chain, you could put there in that in the next blank, knowing that the electron transport chain is a series of redox reactions that releases energy. That energy is used to pump H pluses across the membrane. This is your proton motive force. This means that we have, it's like water behind a dam here. So we have a bunch of H pluses now that now can diffuse back into the mitochondria, changing the shape of the enzyme and making your ATP. All right, and so that's your flow. So this process is not 100% efficient. There's not all the energy in glucose doesn't actually end up with ATP. Where do you suppose some of the energy goes?
Yeah, when you released as heat.
Is released as heat. So that next bullet, I wanted to put that this process is, there's only about 40% of the energy originally in glucose that ends up in ATP. So that first blank for the next bullet is 40%. That might not sound like a lot, but it's actually more efficient than burning gasoline in the car, which is about 25% efficient. So our cells are more efficient, comparatively speaking, than some of the things that we can manually design. So it's pretty efficient compared to other things. And so, so when we talk about energy released here, only about 40% goes to make ATP. The rest of it is released as heat, so about 60%, which means that we are increasing the entropy of the universe, all right, by going through cellular respiration. It's becoming more disorderly as we go through cellular respiration. So therefore, on the second blank in that bullet, so 40% of the energy goes to make ATP. What's our total? 38. So that's what we should put there. Okay. So now let's look at the next
Wait,
picture.
what was the first one?
40%.
Some of you.
And then the second one
look
was
at.
38, she said?
Oh, look. This next picture here. This picture here pulls it all together in one place, and it gives all the totals, how many NADHs and FADH2s and the ATP. But I want to point out some variations that are a little bit different than our calculations. So if we look here, we just said 34 ATP. Notice here, this says 32 or 34. We just talked about how there's 34. And I want to talk about where the difference is between there. So if this is 32, that changes your total. It says 36 or 38. And then if we look at this picture here, there's one thing different, and that's this part right here. This part right here we haven't talked about. So let me explain a few things. Well, first of all, the exact total of ATP is not really known, just in general. All right, so they're still working on studying this and understanding it in more depth. So the exact number isn't known. If you look at another textbook, the numbers will be a little bit different, all right, or could be a little bit different. So let me talk about why our textbook has a variation, 32 or 34 here, and it has to do with the NADH made in glycolysis. This NADH carries 2 electrons. It's out here in the cytoplasm. In order for those electrons in each of those NADH molecules to get to the third step here, to go through the electron transport chain, so it has to get into the mitochondria. Well, guess what? The mitochondria doesn't let NADH in. All right, so therefore, these electrons in NADH are stuck out here. So how do they get here? In comes this electron shuttle. So the NADH gives its 2 electrons, and the 2 electrons move through this protein in the membrane. And depending upon which molecule, you can have a molecule of NAD plus or a molecule of FAD. Those are 2 electron carriers that are naturally found in the mitochondria. And if those 2 electrons go to NAD plus, it forms NADH. If those 2 electrons go to FAD, it forms FADH2. Does that matter? Absolutely. If it goes to FAD, FAD is going to make less ATP, so you're going to get the 32. If those electrons go to NADH, those drop the electrons off at the beginning of the electron transport chain, and you're going to get your maximum amount of ATP, 34. All right. So therefore, it just depends upon which one of those actually get those electrons, because that can't actually enter. All right. And so that's where we get our variations there. And so this process, for our test and for the AP test, you are not going to be asked numbers. So, and that's because they're really not known, all right? So this is, for example, what some people think it's a little bit different. And so what you need to know is the overall process and why oxidative phosphorylation makes the most ATP than citric acid cycle and glycolysis, but I won't ask you how many ATP and things like that, okay, on the test. And so, yeah, they're still actually doing a lot of research with this, and I think now that I show, for simplistic sake, so as the electrons go through, 1 H plus diffuses through for each one of those, and it makes 3 ATP for 1 NADH. They now think that it's possible that there's, as the electrons move through for 1 NADH, 10 H pluses diffuse through, and they're still up in the air about how many H pluses have to diffuse back through to make 1 ATP. Some people think it's 2.5, so it's not even raw, whole numbers, and so on. So they're still researching that, but for simplistic sake, this is what we're going with. All right, and so a maximum of 3 per NADH. All right. Okay, and so so the next section talks about what happens when you don't have oxygen to your cells. So what happens in the absence of oxygen? Somebody tell me why is oxygen present, or why is the presence of oxygen important in this process? What's the role of oxygen, and why is it important? And what happens if you don't have oxygen? What would happen to the process of cellular respiration? So let's start with what its role. What's the role of oxygen in cellular respiration? Role of oxygen. Nathan?
Wouldn't it be the final electron acceptor?
That's exactly right. It's the final electron acceptor. So it's right over here. Here's oxygen. This is where our 6O2s are used. Here's oxygen. It's the final electron acceptor. Without oxygen, guess what? The electrons don't really go through the electron transport chain, which means you don't get the high concentration of H pluses, which means the ATP synthase doesn't get in the right shape, which means that you don't make ATP, which is detrimental for your cells. So that's kind of a snowball effect there without oxygen. So oxygen is very, very important. And so if you don't have oxygen, like if you were to be asked a question like what happens in cellular respiration when you don't have oxygen, and you were just to say that the cell is going to die, you would get 0 points, all right? Because I would expect you to know a little bit more about that, all right? Huh?
If you had the right end answer, would you get like partial credit?
Nah, well, maybe, depending on the wording of the question, all right? But this is an AP class, so I'd expect a little bit more than that, all right? Because, so just to give you an idea, all right? So I just went through that thought process, okay? And so, this is where we're gonna start, all right? We have about 11 minutes, all right? So I'm gonna let you actually get started on your homework, which is the oxidative phosphorylation. first hour had the random fire drill today, and so I'm trying to keep you guys kind of on track, and so you guys get the benefit of starting on your homework. And first hour got to go out in the dark.